The following topics will be covered in this movie clip. First, a comparison between a UV visible absorption spectrophotometer and a fluorometer. Second, a comparison of the structures of chromophores and fluorophores. Third, what are intrinsic and extrinsic fluorophores? Fourth, applications of extrinsic fluorophores in enzyme assays. As shown in this diagram, both UV visible absorption and fluorescence spectroscopies are techniques based on the absorption of electromagnetic radiation by electrons. The two techniques share several common features in their instrumental design. For example, both instruments consist of a light source, a sample holder in the form of a cuvette, and a detector/recorder. The two differ in that, in a UV visible absorption spectrometer, the detector is placed in line with the light source, whereas in a fluorometer, the detector is placed perpendicular to the light source. Molecules that can absorb UV and visible light are commonly referred to as chromophores, and those that can emit fluorescence following absorption are referred to as fluorophores. Comparison of the structure of a chromophore with that of fluorophores indicates that both share several structural characteristics. This is not surprising, because to emit fluorescence, a molecule must first be able to absorb light. In other words, every fluorophore contains a chromophore, but not every chromophore fluoresces. Their structural features overlap significantly, with a few key distinctions that determine whether fluorescence occurs. Structurally speaking, both chromophores and fluorophores generally consist of 1. Aromatic or heteroaromatic ring systems. For example, among the 20 amino acids, only 3, those with aromatic or heteroaromatic side chains, absorb UV light at 200 to 300 nanometers. Aromatic or heteroaromatic ring systems allow electron delocalization and thus lower delta V. For example, an electron at C1 in hexane is localized within its orbital, whereas in benzene, it is delocalized over the aromatic ring. In addition, all 3 aromatic amino acids emit fluorescence after absorption, with tryptophan being the dominant contributor. 2. Conjugated double bonds, or pi systems. Molecules containing alternating single and double bonds enable pi electron delocalization, thereby facilitating pi to pi star transitions. For example, ethidium bromide, dansyl chloride, and Si3 contain both aromatic rings and conjugated double bonds. Thus, all emit fluorescence light following UV absorption. And 3. A nearly planar and a rigid structure. Planar structures maximize orbital overlap and conjugation. Although many molecules with aromatic or heteroaromatic rings absorb light, only a small fraction emit fluorescence upon absorption. For example, although nucleotides such as AMP, GMP, CMP, and TMP absorb ultraviolet light, none appear to emit fluorescence upon absorption. Therefore, a fluorophore is a chromophore with additional structural constraints that favor fluorescence. What is this structural requirement? One of the important structural features of fluorophores is the structural rigidity. Consider a pair of closely related structure, fluorine and biphenyl. Both contain 2 aromatic rings and thus absorb UV light. However, fluorine emits light intensely with quantum yield of 1. In contrast, biphenyl emits light weakly with quantum yield of 0.2. Quantum yield is defined as the ratio of number of photon emitted to the number of photo absorbed. A quantum yield of 1 means that all of the photon absorbed is remitted as fluorescence. A quantum yield of 0.2 means that only 20% of the absorbed photon is remitted as fluorescence. In contrast to biphenyl, in which a freely rotatable single bond connects the 2 phenyl rings, the rigid structure of fluorine restricts vibrational and rotational relaxation, thereby reducing non-radiative decay pathways. Flexible structures of biphenyl absorb light, but energy is often lost as heat or vibration instead of emission. Another example is DNA detection using ethidium bromide staining. Free ethidium bromide in solution has a quantum yield of approximately 0.02 or 2%. Upon intercalation between DNA base pairs, its rotational motion becomes restricted, leading to an increase in quantum yield to 15 to 30%. A comparison of agarose gel staining with ethidium bromide in the presence and absence of DNA shows that a gel without DNA does not fluoresce significantly. However, in the presence of DNA, ethidium bromide binds to the DNA and its motion becomes restricted, resulting in increased fluorescence. Fluorophores have been classified into two groups, intrinsic and extrinsic fluorophore. Intrinsic fluorophores are naturally occurring molecules capable of emitting fluorescence and are found in cellular metabolism. Examples of intrinsic fluorophores include the aromatic amino acids tryptophan, tyrosine, and phenylalanine. Because the fluorescence intensity of tryptophan is sensitive to its environment, the fluorescence emission spectrum of tryptophan in proteins is used as a probe to study protein folding. In contrast to aromatic amino acids, although all four nucleotides absorb UV light, none appear to emit significant fluorescence following absorption. Metabolic cofactors such as NADH/NADPH and F80/FMN are also good fluorophores. In addition, pigments such as chlorophyll A and B are also good fluorophores. Extrinsic fluorophores, on the other hand, are those that are artificially synthesized for diagnostic and research purposes. Examples of extrinsic fluorophores commonly used in the laboratory include dansyl chloride, ethidium bromide, epsilon FNO AMP, and others. Dansyl chloride is used for labeling proteins and nucleic acids, while ethidium bromide is used for staining DNA for detection purpose. Two types of extrinsic fluorophores commonly used for research purposes are single-labeled and double-labeled fluorophores. Examples of single-labeled fluorophores used for enzyme assays are resorphin beta-celebioside and peptide coumarin. Cleavage of the covalent bond by the enzyme releases the fluorophore, resulting in increased fluorescence intensity. When attached to the substrate, the fluorescence of resorphin celebioside and peptide coumarin is quenched by the cellulose or peptide moiety through intramolecular interaction. For instance, if nearby side chain residues of the peptide absorb in a similar spectral region, energy transfer from coumarin to the peptide can occur. A disadvantage of single-labeled fluorogenic substrates is that the addition of a fluorophore often reduces enzymatic cleavage compared to the natural substrate. Therefore, double-labeled fluorogenic substrates have been developed and used in many enzyme assays. In a double-labeled fluorogenic substrate, the substrate is labeled with two different probes, a donor and an acceptor. Furthermore, the donor and acceptor can be labeled at positions far from the cleavage site. Therefore, the addition of a donor and an acceptor does not significantly interfere with the binding of the substrate to the enzyme's active site. Two different donor-acceptor pairs are shown in this diagram. The donor is shown in blue and the acceptor in green. Although not identical, both molecules have similar structures, resulting in overlapping absorption and emission spectra. In an enzyme assay, the fluorogenic probe shown in blue is used as donor, and that shown in green is used as an acceptor. When the donor is excited at a wavelength equal to its lambda max, its electrons are promoted to an excited state. The electrons in the excited state then relax by emitting light. The emitted light has a longer wavelength than the absorbed light. However, if the donor and acceptor are separated by a distance within the Forster radius, typically 20 to 50 angstrom, fluorescence resonance energy transfer, or FRET, can occur between them. Therefore, if the donor is excited at its lambda max and the covalent bond of the substrate has not yet been cleaved, fluorescence resonance energy transfer, or FRET, can occur between the donor and acceptor, resulting in little or no emission. For this reason, the acceptor is often called the quencher. However, upon enzymatic cleavage of the substrate, two products are formed and become separated. Thus, when the donor is stimulated with its lambda max, FRET can no longer occur, leading to increased fluorescence emission.
Bombastic, I like your bum, bum, bum.